Se apresenta,
bom, muito obrigado.
Agora não vai falar, não. Steve. Dá para conectar aí, Marcelo? Tranquilo?
Oi? Bom, professor, muito obrigado pela sua invitação. Meu nome é Fábio. Gostaria que o senhor falasse um pouco sobre situações em que locais muito amplos ou espécies de peixes muito grandes, onde não é possível que a gente faça experimentos no laboratório. Os únicos dados que a gente tem são sobre pesca, por exemplo, em larga escala. Mesmo a coleta experimental é muito difícil. Então, os dados que a gente tem, na verdade, são dados de pescadores ou dados em grande escala, onde nós não temos condições de fazer experimentos controlados. Gostaria que o senhor falasse um pouco sobre o uso de modelos baseados em diferentes situações.
Okay, thank you. That's a difficult question that my fish biologist colleagues and I have just started to think about, mostly because we've been getting questions like this from people in the tropics that are in this situation. On one hand, if you really know nothing, you can't build a model. I mean, it doesn't make sense to build a model based on just some guesses or unsupported assumptions. On the other hand, if any kind of observation or pattern might give you some clues that would help support a model. So I have not had the time to really start reviewing the literature on, for example, the large, diverse tropical fisheries to have any kind of understanding of what we do know and don't know. The kinds of things that we found to be important, one is the physiological metabolic processes. Those are important to link. Well, I should start with what do the fish eat, how do they feed, and how is that affected by environmental conditions? And probably the most important environmental condition is just how much food there is, how much, is there of whatever they eat. And then the physiology helps you convert what you know about their food intake into their fitness, their growth rate, their survival, and their ability to reproduce. So I don't know if there's enough information on some of these fish about these basic processes, what they eat, how they feed, their physiology. Another area that in almost all species we don't know enough is why they die. Biologists like to put lots of emphasis on understanding reproduction, but we know very little, even in species like trout that are the subject of thousands and thousands of papers. There's very few on why they die. So I guess the answer to your question, the best I can say is that we would have to try to assemble what we do know and look for some patterns that we think we could model. And if there are some, then try to build a model that's as simple as it needs to be to try to reproduce those patterns.
Thank you for your marvelous talk. I would like to see if you can push your models for a bit further for evolutionary oriented models. And you showed that when you use alternative behaviors like the night and day strategies for fish, your model gets more complex and predict more variable situations. But animals can change behavior plastically, or they can have variability within the population. And if you have variability within the population, this model could, for example, evolve different lineages, one night forager and one day forager. So what I'm trying to ask you is that if your model could be used to this lineage split to predict things like this or to model things like this.
Okay, thank you. Well, the impolite answer is that what we like to do is just the opposite, which is there was a lot of biologists, trout biologists, believe that the fish in different streams must be genetically different because some feed at night and some feed at day. And so one of the fun reasons we did this experiment was to show that no, this doesn't have to be a genetic difference, that it could just be an adaptive behavior. And I think there's also now laboratory experiments that show the same fish, the same individual, will switch back and forth depending on conditions. So that's actually been one thing that we've enjoyed doing, is showing that these variations even among subpopulations can just be due to plastic behavior, not genetic variability. But individual-based models clearly provide an opportunity for studying the effects of ecology on fitness and evolutionary pressures, and therefore on genetic adaptation. I don't think I'm going to be the person that does that, partly because I don't have any background in genetics, but I would extremely enjoy working with somebody who does have the background in genetics. I think it might be a mistake to, so people have done some simulations using extremely simplified representations of genetic processes. I think there's a small field now called demogenetics or something, where people use individual-based models to look at demography and genetics. I'm only vaguely familiar with that, but this is, I think, an exciting next step. I personally don't know enough to work in that field, but I would love to help someone who does know enough to use our models or their own models to pursue those kinds of questions.
Renata, probably the final question, but let's see how long it is.
Thank you, Steve, for your very nice talk. You showed to us that it's very interesting and possible to use this kind of model to move across levels of organization, and your example was from individuals to populations. And as a landscape ecologist, I think, well, I would like you to comment on how complicated, or if you have experience to crossing from individuals to communities, several populations in especially explicit way, because I think that these are very important for several environmental problems and decisions we have to do. Thank you.
Okay. Let's see. First of all, this trout model that I talked about a number of times, it's actually designed so you can represent multiple trout species. The multiple species generally are quite similar. They might differ only on some characteristics, such as what time of year they reproduce, or so that's a first step. But certainly, yeah, a lot of questions have to do with interactions of a variety of different populations together. I haven't worked with that. I know that there's, I think probably the most prominent example is an extremely large complex model by a person named Christopher Topping, and he's a Danish biologist. I think a lot of scientists think he's extremely ambitious. He's got apparently a very detailed model of an ecosystem in Denmark that includes everything from the plants to the mammals, and I'm not familiar with what all it includes, but as far as I know, it's the most complex attempt to model a whole community. All I can say is that we've learned that every time you add an additional level of either biological organization, or even adding a second adaptive behavior to the model. If you're trying to calibrate it and you reproduce quantitative observations in the real world, it gets an order of magnitude harder to use and harder to understand why you got the result that you did. That's a real trade-off with these models. The potential is endless, but the amount of work that it takes to both build and calibrate the model, but especially to figure out why you got the results you did, also can seem endless sometimes. So there's certainly a potential there. It needs to be explored. It has been to some extent by other people, but it can be quite challenging.
Let me explain why I'm controlling time so strictly. Is because we have 2 coffee breaks to take place at the same time, and we should be first because we don't have enough tables. So very interesting constraint. Danilo.
Okay, thank you. Actually, now and then, when you were responding to Renata, you started responding to my questions.
Go back again.
No, but I was thinking, in my experience of modeling and dealing with IBMs, and yours is much greater than mine. One thing that conceiving the model is not the hardest part, I think, because we look at the system, we get some insights, and we start building a structure and feeding that, changing it until we find it's good. But what I find most difficult is first getting the parameters right, so getting the right value. And within that, then comes the other thing that I find a little bit confusing is that when we build structure, actually analyzing what are the sensibility of the model to varying parameters. So you have 3 or 4 parameters there, and they vary within a certain range. But what will happen to the model is then move these parameters along, and then check the sensibility of the model, and that's always tricky to me. I'm always confused on how to do that. So and I think some people here might think the same. So what would you recommend about the sensibility analysis and how to move with that? Because if you get the parameters a little wrong, it's not a problem, but if they are too wrong, or if a single parameter has a huge effect on the model and the other ones just a slight effect, I don't know how to deal with that so we know we're getting right, so in general.
All right, well, the question is about how to do what we call sensitivity analysis, sensibility analysis. But so how does the model's outcome change if you vary the parameters? And my quick answer is read these books. But no, this trout model that I've talked about quite a bit, we actually had a master's student in mathematics, and that was his thesis, was to do the sensitivity analysis for this trout model. And it was a big project. So it has, maybe 100 parameters that affect very small parts of the model or very big parts. So he varied each parameter over we carefully decided what was a realistic range that each parameter could vary over, and then he ran the model using values over that range. And it was quite interesting. A large number of the parameters had very little effect under the conditions that we simulated, but maybe under other conditions, like if a higher temperature, if it's a parameter that affects temperature, it might become sensitive even though it's not in some conditions. So that was one lesson, that even the results of your sensitivity analysis can depend on the conditions that you're modeling. But we did identify a small number of parameters that were very important. And then looking at the few other individual-based models where people have done this, the most important parameters tend to have to do with metabolism and how much energy the individual needs for a certain amount of growth. Those tend to be very important parameters, and also the parameters for food availability or capturing food. And so once we know which parameters are the most, have the strongest effect, some of those we have pretty good values for because you can measure them in the laboratory. Others are just guesses, and those are the parameters we use to calibrate the model. So another interesting characteristic of this model is that you can calibrate them to a variety of the different outcomes, like the size of the individuals, the number of individuals often are affected by different parameters. So sensitivity analysis can be a big project. That's another reason to keep your model as simple as you possibly can, but also to base your model as much as you can on if there are equations or models for specific processes that have been developed in the laboratory and that have pretty good parameters for them. It might be better to borrow an existing model that's maybe a little more complex than you need, but if it's already a well-studied model, that can be an advantage.
Well, we have a final question by Bruno.
Thank you. I would like to ask about neutral individual-based models. What I've been reading reveals that authors always model individuals looking into the population and not into the individual. Let me explain. I was trying to model how individuals dies in my model, and simplifying it would be simply state a mortality rate, but mortality rate is not a feature of the individual; it's a feature of the population, and I find very tricky to not place my eyes in the population where the individual is important. And in neutral models, where we start to lose the benefits of individual-based models because we are simplifying too much. That's my question.
Okay, first of all, we had exactly the same question about mortality when we're trying to decide, are the parameters that control mortality, is that a habitat parameter or is that an individual parameter? So it's a really interesting question. But when you're, I guess all I can say for an answer is go back to these issues of trying to define a question as clearly as possible and try to reproduce a specific set of patterns as a way to help you answer questions like that. I don't know of anybody that's tried to come up with a clear answer to that question. Let's see, the mortality example is really interesting because in most individual-based models, yeah, there's just an individual parameter for mortality rate, but that doesn't really make any sense. In some models where why individuals die, it's completely unimportant because the model is focused on some other part of the life cycle. In our trout population model, why they die is extremely important because it determines which habitat they use. So a small fish is very likely to die from being eaten by a big fish, so it has to use habitat where the big fish won't go. Whereas the big fish are likely to die being eaten by birds, so they have to go where a bird is unlikely to see them. So in that model, a detailed representation of how the characteristics of both the habitat and the individual affect their mortality risk is important because it drives the key behavior that we're trying to understand, the habitat selection. But again, in lots of other models, if their key behavior doesn't depend on how they die, then maybe there's no reason to represent it in a complicated way.
I'd like to thank Stephen once more. Break until 15:10, até 10h15. Sharp. Porque a gente tem que dar as letras para as pessoas.